Hello, everyone, and welcome to Beyond PCR, Unlocking the Potential of Isothermal Amplification with RCA and RPA. I'm Matt Thorne of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more about our sponsor, please visit thermofisher.com. We do encourage you to participate today, and you can submit any questions you have during those presentations. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You can also submit any technical issues you're having if you're having trouble seeing or hearing the presentation. I'd now like to welcome our first speaker, Rasa, Senior Manager R&D, Molecular Biology at Thermo Fisher Scientific. You can see a complete biography on our speakers on the biography window on the left side of your screen. Rasa, you can now begin your presentation.
Well, thank you for joining. Thank you for nice introducing me. Today, we will present about isothermal amplification as an alternative method to polymerase chain reaction. First, I will start with recombinase polymerase amplification and how it can be used to amplify complex NGS libraries. And later, Raminta will present about rolling circle amplification and mRNA production. Amplification of nucleic acids is at the core of almost all molecular biology workflows because DNA or RNA in the sample can be very precious, limited. Amplification allows to get significant amount of the target region, which is sufficient for further analysis. In detection and genotyping assays, the amplification itself serves as a signal that the target sequence was present in the sample. While in more complex workflows, the product of amplification is checked and taken for further methods for sequencing, for cloning, or in vitro transcription. PCR is as a gold standard of amplification of nucleic acids, and isothermal amplification is getting more and more attention. Why one could choose isothermal amplification instead of PCR? Isothermal amplification has several advantages. First advantage is that equipment needed is extremely simple. Heated water bath is more than enough. Another important advantage is that workflow can take less time, especially if there is a need to get really high amounts of DNA to work with whole genomes, whole plasmids. So isothermal amplification is indeed more efficient. And the last advantage is that isothermal methods are known to tolerate inhibitors present in samples of suboptimal purity. Isothermal means that there is constant temperature, there is no heat to separate DNA strands. For this reason, the polymerases used for isothermal amplification have to be able to separate DNA strands. This activity is called strand displacement. And polymerases that possess strand displacement activity are so-called processive polymerases when they can synthesize fragments without dissociating from the template. There are two types of polymerases used for isothermal amplification. The first is Phe29 polymerase, and its engineered version we have EquiPhe29. And the second type of polymerases is BST-DNA polymerase and other bacterial origin DNA polymerases. The Phe29 is more processive than bacterial polymerases. It can synthesize much longer fragments, and it has another important feature. It has 3' to 5' exonuclease activity. This means that it can correct errors introduced in the sequence, and the product is has the same sequence as the original sample. For this reason, V29 and its engineered versions are used for rolling circle amplification for the methods Raminta will present about later, while BST DNA polymerase is mainly used in the assays the shorter fragments are synthesized, such as the one here. It's called Recombinase Polymerase Amplification. If you look at the scheme is very similar to PCR. It needs only 2 primers and strand displacing DNA polymerases, BST DNA polymerase, with the help of recombinase enzyme. Recombinase enzyme is composed of 2 proteins called UVSX and UVXY. These proteins, in presence of ATP, bind the primers and help the primers to find the complementary sequence in the template. When it's found, another protein, single-stranded DNA binding protein, comes to help. It stabilizes the resulting D-loop structure, and then the extension of the primers can follow. This method is often used to detect pathogens in resource-limited settings, and when there is no lab with thermocyclers, for example. This method is very efficient. It's easy to design primers. It's easy to optimize. It's so robust that we ask the question if it can do something more complicated, if it can amplify libraries for next generation sequencing. To answer this question, we took bacterial genomic DNA from 3 organisms with different GC content, and we did enzymatic fragmentation of the DNA. For RPA, amplicon length is up to 450 base pairs, so we need fragmented DNA, and we ligated sequencing adapters to the fragmented genomes. After this step, we had a very complex mixture of genomic DNA, where all the ends are the same because they are sequencing adapters, and in the middle can be a sequence of almost any GC content. Then we did amplification with primers that are complementary to the sequencing adapters. We did it in parallel using RPA and using specialized PCR master mix for library amplification. And after sequencing, we compared the data obtained after PCR, after RPA, and without any type of amplification, which is the standard, which is gold standard in such experiment because it's not amplified genome. As you can see from the table, in all cases, we got more than 98% of the bases with more than 30 times coverage. We also had good percentages of aligned reads. And when we had a closer look on GC coverage bias, we also found that RPA generates similar profiles as PCR and can be used for amplification of complex libraries. For higher GC content, an additive should be added into RPA reaction to get good coverage bias data. This is everything what we prepared for the best part about RPA. Now I would like to pass the baton to our next speaker, Raminta. She is a scientist at Thermo Fisher Scientific and an expert in rolling circle amplification. Thank you.
Hello, everyone. Thank you, Rasa, for great overview of RPA technology and introducing me. And now I would like to introduce you to another isothermal amplification technique, and this is called Rolling Circle Amplification, or RCA. And this technique is used to amplify circular DNA templates, and it can be performed in two ways. The first way is called linear RCA, and the second multiply primed RCA, or MP-RCA. So in linear RCA, we use a single specific primer, and the design of this primer follows typical PCR primer design guidelines. So you should avoid secondary structures, homopolymer stretches, and repeats. But additionally, as DNA polymerases used in these kind of reactions have strong exonuclease activity, your primer should be exonuclease resistant, with at least 2 phosphorothioate bonds to prevent degradation. So how does linear RCA work? So firstly, primer binds to circular DNA template, and strand displacing DNA polymerase extends around circular DNA. When it reaches newly synthesized DNA strand, it displaces that strand and continues going around the circle repeatedly. This amplification continues in linear manner and results in long concatenameric DNA with tandem repeats of template. In MP-RCA reaction, we use multiple specific or random primers, and the benefit of random primers is that a single set of primers can be used to amplify multiple different DNA templates. The reaction mechanism of MP-RCA is very similar to linear RCA. Firstly, primers bind to the circular DNA template and are extended by strand displacing DNA polymerase. As the polymerase extends the primer, it displaces the newly synthesized DNA strand, which then serves as a template for further primer binding and DNA synthesis. So in this case, reaction continues in exponential manner and results in long, hyper-branched, double-stranded DNA. So if your goal is to produce a single-stranded DNA, go with linear RCA method. But if you need double-stranded DNA, go with MP-RCA. And also, if you need high DNA yields in a short time, MP-RCA is a better option because it follows exponential amplification manner, and that means amplification is faster. You can get higher DNA yields in just less time. So both of these methods uses strand displacing DNA polymerases, such as Fit29 and EquiFit29. Fit29 is highly processive wild-type DNA polymerase with strong strand displacement and exonuclease proofreading activity. The optimal conditions for MP-RCA reaction using Fit29 DNA polymerase is 2 to 4 hours at 30 degrees. But alternatively, you can use EquiFit29 DNA polymerase, and it is Fit29 DNA polymerase mutant developed through in vitro protein evolution. It retains all the benefits of a wild-type enzyme, including high processivity, strong strand displacement, and exonuclease activity. But additionally, it offers improved thermostability, faster reaction speed, higher product yield, and reduced amplification bias. And for reactions using EquiFit29 polymerase, it is recommended to incubate for 1 to 2 hours at 42 degrees. So if your priority is speed and efficiency in your workflow, EquiFit is a preferred choice. So let's look where RCA is used. So RCA is commonly used for two main types of applications. Firstly, it can be used for detection. For example, detecting pathogens like viruses or bacteria, RNA sequences, DNA sequences, even proteins, or genetic variants like single nucleotide polymorphisms or SNPs. Second, RCA is used for DNA amplification before downstream processes. And these can include in vitro transcription, cell-free protein expression, Sanger sequencing, DNA nanoball sequencing, and so on. But today, we are going to explore how RCA can be introduced into workflows for SNP detection, in vitro transcription, and cell-free protein expression. Let's begin by looking at how RCA can be used to prepare templates for downstream applications. So usually, for DNA template for in vitro transcription or cell-free protein expression preparation, we use traditional cloning workflow. And that takes at least 3 days and includes multiple steps like plasmid construction, transformation, colony screening, overnight culture of selected clones, plasmid purification, and this purified plasmid can then be used for downstream applications. So this workflow is labor-intensive, challenging to automate, and have safety risks due to potential bioburden and endotoxin contamination. But these drawbacks can be mitigated by replacing steps involving live cells with a 2-hour RCA reaction. And using RCA in this workflow allows for cell-free, rapid, high throughput, and safer DNA production. So let's analyze this workflow step by step in more detail. So the first step in this workflow is to prepare circular DNA for direct RCA reaction, and that means DNA should be free of nicks. Traditional cloning with restriction enzymes is not ideal for this purpose because it leaves nicks in the final DNA product. So after digesting the backbone and your inserts, the backbone is additionally dephosphorylated, leaving only inserts with phosphoryl groups. And this results in 4 ligation sites, but only 2 phosphates. And that means that 2 nicks are left after ligation reaction. Therefore, this construct is not ideal for direct RCA reaction. But to avoid this issue, we can use alternative method, such as type 2S assembly, also known as Golden Gate assembly. And in this method, both backbone and insert are digested and ligated simultaneously without additional dephosphorylation steps, resulting in fully sealed vector suitable for RCA reaction. Gibson assembly is also an excellent option. This method uses linearized vector and inserts with 20 to 40 base pair overlapping ends. And these fragments are incubated with Gibson assembly reaction mix, and during the reaction, 3 enzyme activities take place. So firstly, exonuclease generates single-stranded overhangs. Complementary overhangs anneal to each other. Then DNA polymerase fills in the remaining gaps, and DNA ligase seals the nicks to generate fully sealed vector, again suitable for RCA reaction. You can also use self-circularization, where the gene of interest is digested and self-ligated into circular DNA without the use of vector. Since vector backbone is not used, this method offers several advantages. Firstly, you have no bacterial elements in your DNA sample, and secondly, you have a higher gene copy number per microgram of DNA, making it ideal for high throughput analysis. So while the last 3 methods, type 2S assembly, Gibson assembly, and self-circularization, can all be used for straightforward RCA template preparation, there is a risk that the products of these assemblies may be not homogeneous, containing both correctly and incorrectly assembled circular DNA, impacting the quality of RCA products. Therefore, the feasibility of Gibson assembly for RCA template preparation was tested by assembling GFP gene into vector with reaction times of 15, 30, and 45 minutes. And as you can see in the graph on the left, there was no difference in the number of transformants between samples, indicating that 15-minute reaction is sufficient. And next, colony PCR confirmed the presence of the inserts in all tested clones, showing that the majority of assemblies are correct and clone selection is not necessary. So then we used 1 microliter of unpurified Gibson assembly products directly for RCA. So no cleanup, no purification, and RCA was efficient with consistent DNA yields. So what does it mean? Basically, you can perform Gibson assembly in just 15 minutes, skip purification, and go straight into RCA. And this makes this whole workflow fast and efficient. However, you should keep in mind that for more complex assemblies, clonal selection may be necessary to ensure correct RCA product formation. Also, we saw consistently high RCA yields across various DNA templates. And these templates included Gibson assembly and self-circularization products, resuspended colonies carrying GFP plasmids, and purified GFP plasmid control as a positive our control. So in all cases, RCA yields reached about 19 micrograms per 20 microliter reaction, while the input was only 1 nanogram of DNA. So to put that in perspective, you would need about 20 PCR reactions to generate the same amount of DNA. So that's quite a lot. And next, the quality and specificity of RCA reactions were evaluated by gel analysis. So undigested RCA products appeared as high molecular weight smear, and that's a clear sign of efficient reaction. And once digested, the resulting bands matched those of digested purified plasmid control bands corresponding to vector backbone and GFP insert. But note that RCA product generated from self-circularization template showed only a single band GFP insert because the vector backbone is not used in self-circularization reaction. So overall, these results confirm that RCA enables high-yield DNA amplification from a wide range of starting materials, whether it's Gibson assemblies, self-circularized products, and even bacterial cells. Additionally, RCA product quality can be confirmed by Sanger sequencing, and again, directly without purification, digestion, or debranching. As you have plenty of DNA after RCA reaction, because you can simply dilute your sample and sequence it. And we have seen high-quality sequenced traces ranging from 900 to 1,000 bases in length from all types of templates. And note that even RCA from resuspended colonies gives great sequencing results. And this means that bacterial genomic DNA present in the colonies does not interfere with plasmid DNA amplification and gives you a possibility to even streamline your traditional clonal selection workflow. So instead of doing colony PCR, when mini-preps, when sequencing, you can simply perform colony RCA and go straight to sequencing. And it can make your clone selection workflow much faster and more efficient. So the next step in the workflow is downstream applications. So let's start with cell-free protein expression. Here, it is important to choose an in vitro translation kit that is compatible with your template's regulatory sequences. If you are working with prokaryotic templates, use a prokaryotic kit that supports coupled in vitro transcription and translation. A good example of such a kit is in vitro Gen Expressway mini cell-free expression system. So to test this, we use just 1 microliter of various RCA products as input and monitor GFP synthesis by measuring fluorescence every 5 minutes. So apparently, RCA products enable cell-free protein expression of GFP just as efficiently as purified plasmid control. So it shows that RCA products without any additional modifications can be used as template for protein expression. So the next application of RCA products is in vitro transcription. In the gel image on the left, you can see RNA synthesized from two types of DNA templates: linearized purified plasmid DNA and digested RCA product cut to match linearized plasmid, which encodes about 1 kilobase long messenger RNA. So in both cases, we see a single band of the same size, confirming that RCA products support the synthesis of correctly sized messenger RNAs. And the transcription yields were also comparable, reaching about 120 micrograms. And this shows that RCA can be used to efficiently generate templates for IVT reaction. What is even more interesting, that we can perform this workflow in a single tube. So firstly, we can amplify plasmid DNA via RCA reaction when restriction reagents are added directly into the same tube, and after digestion, IVT reagents are supplemented. So this sequential process, RCA, digestion, and IVT, from just 1 nanogram of plasmid DNA can yield over 1 milligram of RNA. In comparison, to achieve the same RNA yield with a purified plasmid template, you would require 10 micrograms of that plasmid input. So it reduces the amount of required template. But it not only reduces the required amount of template, it also speeds up the workflow and offers potential for automation. So RCA is a powerful method to prepare DNA templates for applications like protein expression and in vitro transcription. And by replacing traditional cloning steps that can take about 2 days with a single 2-hour RCA reaction, the workflow is reduced from 3 days to 1 day. And additionally, being entirely cell-free, there are no endotoxins, bioburden, or E. coli-related issues. And we believe that RCA will be used daily to speed up high-throughput research like protein screening in the future. But RCA isn't just for DNA template preparation. It's also effective for detecting pathogens, RNA, genetic variants like SNPs, the most common type of genetic variation in humans. And SNPs occur approximately once every kilobase, meaning there are roughly 5 million in human genome. And while the most of them are neutral, some can influence how individuals react to diseases, drugs, vaccines, and other factors. Thus, SNPs can serve as important biomarkers, and they are analyzed using various methods. And notably, they can be detected through workflow, which includes ligation followed by RCA reaction. And here is how it works. So we begin by designing 2 specific oligonucleotides called padlock probes or PLPs. And one is specific and complementary to the wild-type SNP allele, and the other to the mutant allele. So the specificity of each padlock probe is defined by its 5' and 3' ends, which must be fully complementary to the SNP region of interest. So let's say we're analyzing a SNP locus that can contain either C or G, as shown in the figure. Imagine the sample is homozygous wild-type. And this means that wild-type specific padlock probe will be fully complementary and anneal to the target sequence, forming an open circular structure. Because ends are perfectly matched with the target, DNA ligase joins these ends into a closed circle. Any unligated linear DNA is then removed by exonucleases 1 and 3, and the remaining circular DNA is amplified via Rolling Circle Amplification, RCA, and reaction products can be detected using molecular beacons, CRISPR-Cas detection systems, nucleic acid stains, and more. On the other hand, if PLP has even a single base mismatch, as with mutant-specific padlock probe in this example, circularization does not occur. Mismatched PLPs are degraded, RCA is not initiated, and no signal is detected. Now, if you have homozygous sample for mutant allele, the opposite happens. Only mutant-specific padlock probe will be ligated and will produce a signal. And lastly, if a sample is heterozygous, both wild-type and mutant-specific padlock probes will produce RCA signals. However, pay attention that reliable SNP detection using this workflow depends on DNA ligase we use. It must be high-fidelity DNA ligase and efficiently ligate fully complementary probes while rejecting those with even a single base mismatch at ligation site. So to find the best ligase for SNP detection, we tested the fidelity of 3 thermophilic DNA ligases. And as shown in the figure on the left, all 3 discriminated well against double mismatches, while only a few was ligated. However, mismatch position matters. Fidelity was lowest with mismatches at 5' end, while 3' mismatches were more reliably rejected. Therefore, SNPs should be positioned to be complementary to your to the 3' end of your padlock probe. And among the ligases we tested, thermophilus DNA ligase showed the highest fidelity, with no ligation for double mismatches and lower efficiencies for both 3' and 5' mismatches, making it the most reliable choice for high accuracy SNP detection research. And now, look at the figure on the right. You can see how we optimized ligation accuracy by testing thermophilus DNA ligase at higher reaction temperatures or with shorter hybridization regions. And increasing the temperature from 55 to 17 degrees Celsius or reducing hybridization regions from 24 to 14 nucleotides significantly improved specificity. Under these conditions, matched substrates were ligated efficiently, while mismatched ones had a ligation efficiencies below 10%. So in summary, for SNP detection, we recommend to use thermophilus DNA ligase, position SNPs to be complementary to the 3' end of your padlock probes, and apply higher reaction temperatures or shorter hybridization regions. So you should design your padlock probes with shorter hybridization arms. So using these optimized conditions, we perform SNP detection workflow with thermophilus DNA ligase for detection, Equifeed 29 DNA polymerase for MP-RCA reaction, and picogreen nucleic acid stain for real-time detection of amplified products. And as it was highly specific, no signal was detected for non-specific padlock probe and target combinations. And for matched pairs, the time to signal varied with input amount, with higher input leading to a faster signal. What is really exciting about this method is that this method isn't just highly specific, but it is also compatible with multiplexing. And now there are even advanced techniques based on ligation and RCA workflow that allow the detection of up to 100 SNPs at once. And this takes biomarker analysis to entirely new level, and I'm really excited to see the future of this research and progress. And now I would like to jump a little bit to another topic and explain how multiple displacement amplification can be used to amplify whole genomes. So MDA is used to amplify DNA non-specifically, and it follows the same principles as we have already discussed for MP-RCA reaction. So it uses the same strand displacing polymerases, it uses random primers, and it uses the same mechanism. Just to remind you, random primers bind along the template, and DNA polymerase extends these primers. And as it goes, it displaces the strands ahead of it, creating new single-stranded regions where more primers can bind. So this results in hyperbranch structure, and you can end up with even DNA fragments reaching 100 kilobases. So MDA can amplify entire genomes from really tiny amounts of template, even from a single cell or just picograms of DNA input. And that's why it's a popular method for whole genome amplification. When we are working with limited DNA samples and want to increase available DNA to make it sufficient for our needs. And this can be crucial in areas like pre-implantation diagnostic research, forensics, single-cell sequencing, SNP genotyping, metagenomic studies, cancer research, and so on. And this whole genome amplification using MDA is very simple. It follows a straightforward three-step workflow. First, cells are lysed and DNA is denatured. Then MDA using Equify-29 DNA polymerase amplifies the entire genome in just 1 hour. And finally, a sequencing library is prepared for NGS. And so this entire process for DNA pre-amplification before library preparation takes about 2 hours with only 30 minutes of hands-on time. But to ensure high-quality data, whole genome should be amplified with even coverage and minimal bias. And to assess these features, we tested MDA using different strand displacing DNA polymerases. And this slide shows the results. So firstly, take a look at the graph on the left. So it shows NGC bias from DNA pre-amplification of mixed bacterial genomes with low, medium, and high GC content. And these pre-amplified samples were sequenced on Illumina platform, and the coverage was normalized to reveal GC bias. Ideally, a bias-free reaction would result in a flat line at 1 across all GC percentages. The green line here representing an amplified control DNA comes closest to that ideal. Not far behind is a yellow curve, and this is a sample pre-amplified with Equify-29 DNA polymerase, which stands out for its low GC bias, outperforming other polymerases tested. And now look at the graph on the right. So here we are looking at the coverage evenness of single-cell genomes that were pre-amplified using MDA reaction. And ideally, we want every base to be sequenced with the same coverage, a perfect score of 1. But as we know, nothing is perfect in the real world, and our control and amplified genome, labeled in green, had evenness score of 0.75. So the next best score of 0.5 came from Equify-29 DNA polymerase, demonstrating that Equify-29 is the best option for whole genome amplification reaction. So generally, MDA allows us to amplify DNA from incredibly small amounts. You can think of a hair follicle or a tiny blood stain, and it allows us to amplify it with a good coverage, evenness, and low GC bias. So this opens up huge possibilities for us, from solving crimes with limited genetic material to studying the genetic makeup of various organisms in our environment. So with that, I would like to conclude our seminar. And today we have explored how isothermal amplification can simplify research. We have covered a bunch of topics, including RPA for NGS library prep, RCA and MDA for cell-free protein expression, SNP genotyping, whole genome amplification, in vitro transcription. But if you are interested in learning more about isothermal amplification techniques, be sure to visit our website for more information. And now I would like to thank you all for joining us today. A big thanks to LabRoots for hosting the webinar, And now I think we are ready for our Q&A session, right?
Yes, so thank you very much, both of you, for your very informative presentations. Before we jump into the Q&A, we do have a quick poll we'd like to put out there. How can Thermo Fisher Scientific support your research needs in isothermal amplification technology? If you'd like to look those over and submit your answer there. While we're waiting on those answers, I'd like to point out also on the left side of the screen is a survey for the webinar. It will also pop up after the webinar is complete. So if you haven't submitted your questions for the Q&A, you can do so as well. Put those into the Q&A box, and then you can hit submit, and we'll try to get to as many of those as we can. We'll just give the poll just another moment or two, and then we'll jump into those questions. We'll go ahead and start with that first question while that poll finishes up. The first question is for Rasa. Can RPA also be used on complex genome, human and large plant genomes?
So hello again, great question. Yes, it can. Three bacterial genomes, it's a very good model system to look for GC coverage, but the main target, of course, is to use it on complex genomes, libraries for complex genomes.
All right, thank you. The next one we have is for both. How about specificity and sensitivity in isothermal amplification compared to real-time PCR?
Well, everything depends on assay design, on primers, so it can be a lot of cases. In general, yes, you can reach similar sensitivity and specificity using isothermal methods as PCR. Maybe Raminta has something to add.
I want to point that RCA reaction usually is used to amplify everything that you have in the sample, and it is not directly created to amplify specifically DNA fragments. But if you design your primers to be specific to the template, the specificity is similar to real-time qPCR reactions.
All right, thank you. This one is for Rasa as well. Are there possibilities to generate longer amplicons greater than 1 kilobyte?
With RPA, unfortunately, no. PCI is a technique that can amplify a larger fragment. However, if going to RCA, you can generate even larger fragments than with PCR.
All right, thank you. Let's see, the next one we have here, Ramenta, what are the rules or guidelines for designing padlock probes? Oh, you're still on mute there.
Okay, so now you can hear me. So there are actually no strict rules for padlock probe design. I can just give you some guidelines. So these padlock probes are usually between 50 and 150 nucleotides, but mostly I see in the literature that the padlock probe is about 70 nucleotides long. And these padlock probes consist of three main parts. So the 5' end, the middle region, which is called often like a spacer, and 3' end. So these 5' and 3' ends are specific to the target, and they are designed to be complementary to adjacent sequences on the target nucleic acid. And after annealing, they form like open circular structure. And this hybridization region should be between 20 to 30 nucleotides long, But as I already discussed in our webinar, it can be changed. So in our SNP detection workflow, we changed these hybridization regions from 24 to 14 nucleotides. So there are no strict rule. And now about middle region with spacer. It should be free from complementary sequences to our target DNA or any other sequences to avoid non-specific binding. And additionally, it's important to ensure that both the spacer and ends of a probe do not form stable secondary structures, which can interfere with hybridization and ligation process. But as I mentioned, there are no strict rules how to design padlock probes, and often you can find different modifications of padlock probes in the papers.
All right, thank you. Another one for you. In several cases during the webinar, it was mentioned that RCA products can be used directly for downstream applications. Are there any downstream processes that require RCA product purification or modifications?
Thank you for the question. Yes, so typically RCA products can be directly used in downstream applications without cleanup, but of course there are some that require modifications. So for example, RCA product should be purified before quantification if you want to measure concentration by measuring absorbance or by nanodrop, because DNTPs, primers can interfere with concentration measurements. So in this case, I would recommend to purify your RCA products using magnetic beads based separation and purification. But on the other hand, there is a possibility to quantify RCA products directly using picogreen double-strand DNA assay or using qubit fluorometer. Also, sometimes hyperbrain structure is not the best for your downstream applications. It can be in the case of nanopore-based DNA sequencing. So before nanopore-based DNA sequencing, the branching is required of RCA products, and you can perform it using S1 nuclease. And we have a protocol for it in our EquiV29 DNA polymerase manual. And digestion of RCA products can also be beneficial. For example, if you are doing IVT reaction, your RCA product should be digested into 1 repeat sequences to ensure that your RCA reaction products are correct length. So yeah, there are several modifications, but basically you have to think whether the structure or components of reaction can somehow interfere with your reaction. And if it is so, you can do either purification or the branching or digestion of your RCA products.
All right. One more here for you. Could you make a comparison of the amplification specificity of those above isothermal amplification method, RPA, RCA, MP, RCA, MDA?
MDA is like a basic method, and these RCA methods and MPRCA, linear RCA, and MPRCA, and whole genome amplification uses MDA. In linear RCA, we are using only single primer. So the product is linear, and it is not hyperbranched. It contains many repeats of the same sequence. While in MPRCA, we have a hyperbranch structure. We are using multiple different DNA primers. And usually, with RCA reaction, whether it's linear RCA or MPRCA, we want to amplify everything that we are having in our sample. While RPA is more used for amplification of targeted sequences. So it's like a similar to PCR reaction. Yeah, I guess that's it.
Okay. For Rasa, how many regions in human genomic DNA can be designed for the multiplex PCR reaction?
Multiplex PCR depends. We are talking about endpoint PCR or quantitative PCR. Quantitative PCR multiplexing is limited by amount of channels we can detect, but there are assays where hundreds of regions in human genomes can be amplified for endpoint detection. RPA is a similar method to PCR, so it can also be used for multiplexing from genomic DNA. The main thing to consider when multiplexing is primer design, because the more primers you have, the more interactions we can have between them. And also if working with RPA, primers should be longer than for PCR. So it's not the same primers that work with PCR cannot be used with RPA in some cases.
Another one here for you. Let's see. Can multiplex possible with isothermal amplification? Could thermo RPA kit reach so far in your own study?
In our study, we currently amplified up to 6 amplicons at the same time with RPA, but in general, it's possible to do even more. RPA, it's a great method for multiplexing because it needs only 2 primers if compared to other isothermal methods such as LAMP, for example. So in LAMP, you need 6 primers, so it's much more difficult to design multiplex assays. So from isothermal methods, RPA is the method of choice for multiplexing.
All right. Another
and
one.
Of course, I can add. Raminta also commented about RCA, but it can be used for multiplexing.
Yeah. Another one for Raminta. How important is temperature stability in the instrument profile during the amplification process in the machines that you mentioned? And if yes, how stable is the temperature reservoir?
So for isothermal amplification, I mentioned several methods. So it's RCA, MDA, and MPRC, and they all use similar ranges of temperature. So if you're using Fe29 DNA polymerase, the best temperature is 30 degrees, but it can range between 25 to 37 degrees. And for Equife29 DNA polymerase, the optimal temperature is 42 degrees, but it can vary between 37 to 45 degrees. So the range is quite big, so it's not so important to have a stable temperature in the instruments you use.
All right, thank you. Next one for Rasa. Do you have the data on plant or seed pathogen detection assays using isothermal amplification techniques?
Yes, we do. We have application notes using LAMP technique for plant pathogens, so we can find them in our webpage.
All right. Another one for you, Rasa. What is the preferred technique for the pathogen detection, LAMP or RPA? And what conditions should be considered when choosing between the two?
Both are widely used and very efficient for detection. For LAMP, as I already mentioned, you need 6 primers. So LAMP assays are more difficult to design, and for some pathogens, maybe it's complicated and you need some bioinformatics analysis for LAMP. But it has, if you have well-designed LAMP primers, specificity is really great. RPA from the back end, it's very easy to design. It's only 2 primers. So this is and it's the main advantage. Also, it can be combined with other techniques to increase specificity, such as CRISPR-Cas systems.
All right. The next one here, Ramenta. For MDA, when handling small volume samples, is nucleic acid isolation required before amplification?
So in the standard MDA workflow, it is not required to isolate nucleic acids before amplification, especially when you are using cells that are easily lysed. And this lysation step is fully sufficient for amplification. However, if you are working with cells that are more difficult to lyse, such as those with thick cell walls, DNA isolation may be necessary to ensure successful amplification. And if you are performing MDA reaction without any purifications, but you have problems with it, I would suggest you purify the samples as a troubleshooting step, because maybe you have inhibitory components in your reaction, and this can help eliminate that and improve your success of MDA reaction. But in general, it is not required to purify your DNA samples.
All right. We do have time for a few more questions here. Rasa, one for you. Isothermal amplification reaction temperatures are much lower than PCR. How does this address non-specific amplification challenges?
So a lot of, again, depends on primer design. But LAMP, for example, with 6 primers and quite high temperature, if well designed, can be really specific. RPA primers are also longer, so this when for PCR, so this allowed to have similar specificity as in PCR. Also, it can be increased by combining with CRISPR-Cas systems and similar helpers.
One more for you. Do isothermal amplification techniques work for RNA-specific amplification, such as RNA virus genome sequencing or amplification?
So RPA can be combined with reverse transcriptase, same as with LAMP. If speaking about virus sequencing, where there is a need to amplify the whole virus genome, I would recommend one-step RT-PCR.
Looks like we've got time for one more. So, well, Rasa, what are the critical factors to consider when designing primers for RPA, RCA, or MDA, and how do they differ from traditional PCR?
So for RPA, you have to consider same things as for PCR primers. So primers should be specific for the target region. They should not form primer dimers or secondary structures. And a very important thing for RPA is the length. Primers should be longer than 30 bases. This is critical. For techniques that use feed 29 polymerase, the critical thing is to make sure that primers are not degraded by polymerase and they have PTO bonds at 3'. The same is true for specific primers, for rolling circle amplification, and for random primers as well.
All right. Well, I'd like to thank both of our presenters for their time and their very important research. I'd like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. And before we go, I'd like to thank the audience for joining us and all the questions. Any questions we didn't have time for and those that get submitted during the on-demand period will be submitted to our speakers. So please take the time to complete the survey that'll pop up after the webinar is complete. And this webcast will be available on demand, and LabRoots will alert you via email when it's available for replay. We do encourage you to share that email with your colleagues who might have missed the live event today. So until next time, thank you very much and goodbye.